Welcome back to the MedPod. You know, last time we talked about how biophysics is like this amazing tool in medicine,
Yeah.
but this time we're going deeper,
Right.
like way deeper into the
Yep.
like biophysical principles
Yeah.
behind some of the most like fundamental processes
Yeah.
that are happening in your body every second.
It's amazing.
Ready for like a deep dive into
Let's do it.
sedimentation,
Sure.
electrophoresis, biological membranes, and even like how your muscles work.
Absolutely. It is fascinating how physics and chemistry
Right.
literally shape the processes of life
I know.
down to the cellular level.
It's mind-blowing.
It is.
Okay, so let's unpack this.
Okay.
Our first stop is
Okay.
the lab.
Right.
Imagine you're like a researcher, and you're trying to isolate a specific protein
Yeah.
from a complex mixture.
Yeah.
How do you even begin
Right.
to separate these microscopic components?
Well, you could start with a technique like sedimentation. It's like using this superpowered centrifuge
Oh, wow.
to separate particles
Okay.
based on their size and density. Think of it like spinning a salad spinner.
Uh-huh.
The heavier ingredients get flung to the edges while the lighter ones stay closer to the center.
So heavier, denser particles end up at the bottom of the test tube.
Exactly. And for even more refined separation,
Oh, right.
scientists use something called density gradient centrifugation.
Okay.
It's like creating a layered cake in the test tube,
Oh.
with each layer
Okay.
having a slightly different density.
Got it.
This lets researchers pinpoint and isolate molecules with very specific characteristics,
Yeah.
like separating different types of DNA or proteins.
So it's like a microscopic obstacle course for
Yeah.
molecules, with each one finding its place in the density gradient.
Precisely. Now, electrophoresis
Okay.
is a different but equally important technique. Instead of density,
Okay.
it uses an electric field to
Right.
separate molecules based on their size and charge.
Oh.
Imagine a microscopic racetrack
Okay.
with a positive end
Yeah.
and a negative end. Molecules with a negative charge will race towards the positive end and vice versa.
So smaller, more highly charged molecules would zip across the finish line first.
Exactly. And this is how DNA is analyzed
Wow.
in forensics, or how proteins are separated in blood samples to diagnose diseases.
That's incredible.
There are even more specialized types of electrophoresis,
Okay.
like SDS-PAGE, which
Sure.
separates proteins primarily based on their size,
Uh-huh.
and isoelectric focusing,
Okay.
which separates them based on their charge.
So sedimentation and electrophoresis are like the dynamic duo
I like that.
of biophysical techniques,
Yeah.
helping us sort through the incredible complexity
Yes.
of biological samples. But let's shift gears
Okay.
and zoom in on one of the most fundamental
Yeah.
structures in your body,
Yeah.
the cell membrane.
Oh, the cell membrane is fascinating.
I'm a nerd.
It's more than just a barrier between the inside and outside of the cell.
Right.
It's a bustling hub of activity,
Yes.
constantly regulating what goes in and out.
Right.
And its structure
Yes.
is key to its function.
Wait, remind me about the structure of the cell membrane again. What is it made of?
It's primarily composed of a phospholipid bilayer. You remember phospholipids, right?
Yes.
They have these hydrophilic heads that love water
Uh-huh.
and hydrophobic tails that avoid it. In the cell membrane, 2 layers of phospholipids arrange themselves tail to tail,
Right.
forming sort of a sandwich with a water-loving exterior
Okay.
and a water-fearing interior.
Oh, right.
Yeah,
That's why it's called a bilayer.
exactly.
It's not just phospholipids, right?
Correct. Embedded within this bilayer are proteins that act like gatekeepers,
Oh, wow.
channels, and even sensors. This mosaic of phospholipids and proteins
Okay.
is constantly in motion,
Mhm.
shifting and adjusting.
Got it.
Hence the term fluid mosaic model.
Okay, so the cell membrane is like this dynamic city wall,
Yes,
selectively letting certain molecules in and out.
exactly.
But it's also involved in creating an electrical potential across the membrane.
Well,
Right?
exactly.
That's the resting membrane potential.
You see, the cell membrane doesn't allow ions to freely pass through.
Okay.
It carefully controls their movement,
Right.
creating an imbalance of charges between the inside and outside of the cell.
So it's like a tiny battery
Yes,
powering all the amazing processes happening inside the cell.
a perfect analogy.
Wow.
The key players here are sodium and potassium ions.
Okay.
There's a higher concentration of potassium inside the cell and a higher concentration of sodium outside. This difference in concentration, combined with the selective permeability of the membrane, creates an electrical potential.
But how is this imbalance maintained? It seems like the ions would just want to even out over time.
That's where specialized proteins called ion channels and pumps come into play.
Okay.
These proteins are embedded in the membrane
Yeah, okay.
and act like gatekeepers, selectively allowing certain ions to pass through.
Oh,
And there's a particularly important pump,
okay.
the sodium-potassium pump,
Yeah.
that works tirelessly to maintain this uneven distribution.
So it's like a constant tug of war
It is.
with these pumps working against the natural tendency of ions to diffuse evenly.
Exactly.
And this difference in charge, this
Right.
electrical potential, is what we call the resting membrane potential.
Exactly. And this potential is crucial
Okay.
because it acts like a coiled spring, storing energy that can be released to power
Oh,
various cellular processes.
so it's like potential energy.
Yes. In fact, the resting membrane potential is the foundation for how your nerves transmit signals.
Nerves.
Yes.
So we're going from cell membranes.
It all ties together beautifully.
Oh, wow.
You see, your nerves use electrical signals
Okay.
called action potentials to
Get it.
transmit information throughout your body.
Uh-huh.
And those action potentials are rapid changes
Okay.
in the resting membrane potential.
Okay, I'm intrigued. How do these action potentials work?
Think of your sensory receptors
Okay.
as highly specialized antennas,
Right.
each tuned to a specific type of stimulus.
Okay.
Light, sound, pressure, chemicals, you name it.
Right.
When a stimulus is strong enough to activate a receptor,
Okay.
it triggers a cascade of events
Okay.
that leads to the generation of an action potential
Huh?
in a nearby nerve cell.
So it's like a domino effect.
Precisely.
Starting with the sensory receptor
Yes.
and ending with a signal being sent
Right.
along the nerve.
No, the action potential itself
Right.
is a rapid change in the membrane potential
Yeah.
that travels down the nerve fiber like a wave.
Oh, wow.
It involves a rapid influx of sodium ions into the cell,
Okay.
followed by a rapid outflow of potassium ions.
Okay.
This cycle of depolarization
Alright.
and repolarization
Alright.
is what propagates the signal
Okay.
along the nerve.
It's like a carefully choreographed
Yeah.
dance of ions,
It is.
creating a wave of electrical activity that
Yes.
zips down the nerve fiber.
Exactly. And the speed and efficiency of this signal transmission is mind-boggling.
I know.
It's how you can react to stimuli in a fraction of a second.
Right. Like, that's so crazy.
It is.
So from feeling a pinprick
Yeah.
to seeing a flash of light,
Yeah.
it's all thanks to these rapid changes in the resting membrane potential.
Exactly.
It's amazing how much is happening at the cellular level,
It really is.
all orchestrated
It is amazing.
by the physics of ions
Yeah.
and membranes.
Truly is. And speaking of amazing things happening at the cellular level,
Yeah.
let's talk about how your muscles contract.
Okay. Let's get into it.
It's another example of biophysics in action,
Yeah.
but this time we're zooming in on the molecular machinery
Okay.
that powers movement.
Muscles. Okay, but I need a minute to picture my muscles as something more than muscles.
Right.
What's a good way to visualize how they work at the microscopic level?
Imagine your muscles as a collection of tiny, incredibly sophisticated engines.
Okay.
Each engine is a sarcomere,
Yeah.
the basic contractile unit of muscle.
Okay.
And within each sarcomere,
Uh-huh.
you have two key players, actin and myosin.
Actin and myosin. Those are proteins, right?
Yes. Actin forms thin filaments,
Okay.
and myosin forms thick filaments.
Got it.
These filaments are arranged in a very specific overlapping pattern
Uh-huh.
within the sarcomere.
Okay, so we've got these filaments arranged like a microscopic assembly line.
Yeah.
How does that lead to contraction?
Well, myosin has these little heads
Okay.
that can bind to actin,
Uh-huh.
and with the help of APP,
Yeah.
the energy currency of the cell,
Right.
these myosin heads pull on the actin filaments,
Okay.
causing them to slide past each other.
So it's like a microscopic tug of war
Precisely.
with myosin pulling on actin,
Yes.
causing the sarcomere to shorten.
And when millions of sarcomeres shorten simultaneously,
Oh, wow.
the entire muscle contracts,
Yeah.
generating force and movement.
So it's like a perfectly synchronized dance of molecules
Yeah.
powered by ATP.
Yes. But how is this contraction controlled?
Right.
Do our muscles just contract randomly?
I was just thinking that. Like, how does it know when to do this?
Not at all. There's a sophisticated regulatory system in place.
Okay.
Two key proteins,
Uh-huh.
tropomyosin and troponin,
Okay.
play a crucial role. Tropomyosin is like a rope that covers the binding sites on actin,
Okay.
preventing myosin from attaching
Uh-huh.
when the muscle is at rest.
So it's like a safety mechanism,
It is.
making sure that the muscles aren't contracting
Exactly.
all the time.
But how does the muscle get the signal to contract when it needs to?
Right. Exactly.
That's where troponin comes in.
Okay.
It's a complex of 3 proteins
Okay.
that binds to both tropomyosin
Right.
and calcium ions.
Okay.
When a nerve signal triggers the release of calcium ions inside the muscle cell,
Yeah.
they bind to troponin,
Okay.
causing a conformational change
Got it.
that moves tropomyosin out of the way,
Let's go.
exposing those myosin binding sites on actin.
So calcium is like the key that unlocks the muscle contraction.
Exactly.
It's fascinating how this intricate
It is amazing.
dance of proteins, ions, and energy
Yeah.
allows us to move with
Mhm.
precision and power.
It truly is remarkable. And it highlights how biophysics reveals the elegant interplay of structure and function at the molecular level. But muscle contraction isn't the whole story.
Oh.
To truly understand how our muscles work, we need to delve into their mechanical properties.
Mechanical properties. Okay, I'm ready to dive into that. But first, can we take a quick pause? I need to grab some water.
Of course, take your time.
I'm getting thirsty from all this brain-bending biophysics.
We'll pick up our exploration of muscle mechanics when you're back.
Okay.
Welcome back, ready to delve into the mechanics of muscles.
Definitely. I'm imagining my muscles as these like tiny engines.
Yes.
But like those engines need to be controlled, right?
Mhm.
How does my body fine-tune the force
Mhm.
of a muscle contraction?
Yeah.
I mean, picking up a pencil uses way less force
Yeah.
than lifting a heavy weight.
That's where the different types of muscle contractions come in.
Okay.
Twitch,
Okay.
summation, and tetanus.
Okay. Break those down for me. What's a twitch?
A twitch is the simplest type of contraction.
Okay.
It's a quick, jerky response to a single stimulus,
Okay.
like a brief electrical zap to the muscle.
Uh-huh.
Imagine tapping your finger on a table.
Okay.
That's essentially a series of twitches.
Got it. So what about wave summation?
Yeah.
Does that have something to do with
Yeah.
multiple stimuli?
You're exactly right.
Okay.
Wave summation occurs when a muscle is stimulated repeatedly in quick succession.
Okay.
Each stimulus builds upon the previous one,
Got it.
leading to a stronger, more sustained contraction.
So it's like sending multiple signals to the
Yeah.
muscle in a row.
It is.
Telling it, keep contracting, keep contracting.
Precisely. And if the stimuli are frequent enough,
Yeah.
the muscle reaches a state of tetanus.
Tetanus, isn't that like a dangerous infection?
Yes, there's a disease called tetanus.
Right.
But in this context, we're talking about a physiological state.
Okay.
In tetanus, the muscle is in a state of maximal sustained contraction. It's what allows you to hold a heavy object steady or maintain a certain posture.
So it's like holding a plank.
Yeah,
Your muscles are in a state of tetanus,
exact.
constantly firing to keep you in that position.
Now,
Yeah.
beyond the types of contractions,
Yeah.
there's another fascinating aspect of muscle mechanics,
Okay.
the length-tension relationship.
Okay. What's that all about?
Imagine stretching a rubber band.
Okay.
If it's stretched too little,
Yeah.
it won't generate much force when you release it.
Uh-huh.
But if you stretch it too much,
Yeah.
it might snap.
Right.
There's a sweet spot where the rubber band can generate the most force. Muscles are similar.
So there's an ideal length
Exactly.
for a muscle to generate maximum force.
It all has to do with the overlap
Okay.
between those actin and myosin filaments we talked about earlier.
Mm-hmm.
There's an optimal overlap
Okay.
where the myosin heads can bind to the most actin binding sites,
Okay.
leading to the greatest force production.
That makes sense. But what about speed?
Okay.
How does the speed of a contraction affect the force a muscle can generate?
That's described by the force-velocity relationship.
Okay.
Basically, as the speed of a contraction increases,
Okay.
the force it can generate decreases.
So it's like trying to push a heavy object.
Yeah.
You can move it slowly
Exactly.
with a lot of force, but if you try to push it quickly,
Right.
you won't be able to generate as much force.
And this relationship is important for understanding how muscles perform different tasks.
Okay.
For example, when you're lifting a heavy weight,
Yeah.
your muscles contract slowly to maximize force production.
Okay.
But when you're sprinting,
Uh-huh.
your muscles contract quickly, sacrificing force for speed.
This is so cool.
It is!
But muscles don't work in isolation, do they?
No.
They're attached to bones.
Right.
And those bones act as levers.
You're absolutely right.
Okay.
Our bodies are incredible examples of biomechanics in action. Our muscles act as the force, our bones act as levers,
Okay.
and our joints act as fulcrums.
Okay, I'm remembering those lever systems from
Yeah.
physics class.
Yeah.
But can you remind me how they work in our bodies?
Think of a seesaw.
Okay.
The fulcrum is the pivot point in the middle.
Yeah.
The closer the weight is to the fulcrum, the easier it is to lift.
Right.
That's a mechanical advantage.
Okay.
But if the weight is far from the fulcrum,
Yeah.
it's much harder to lift. That's a mechanical disadvantage.
So some levers in our bodies are designed for power,
Precisely.
while others are designed for speed or range of motion.
Our bodies are full of different lever systems,
Oh, wow.
each optimized for specific tasks. The biceps muscle in your arm,
Okay.
for example, acts as a third-class lever,
Okay.
where the force is applied between the fulcrum, your elbow,
Right.
and the load, whatever you're lifting. This arrangement
Okay.
gives us a mechanical disadvantage,
Okay.
but allows for a wide range of motion.
So I'm trading power for flexibility with my biceps.
Exactly.
Interesting. What about other muscles?
Well, take the gastrocnemius muscle in your calf.
Okay.
It acts as a second-class lever,
Okay.
giving us a mechanical advantage
Why?
for powerful movements like jumping.
It's amazing how our bodies have evolved to use these lever systems so efficiently.
It is.
But muscles aren't the only tissues
Right.
with important mechanical properties, are they?
You're absolutely right. Let's shift our focus to the broader world of tissues,
All right.
where mechanics plays a critical role
Yep.
in everything from bone strength to blood vessel elasticity.
Okay, I'm ready to stretch my mind and learn about the mechanical properties of tissues.
Okay,
But where do we even begin? It seems like a pretty broad topic.
well, let's start with a simple analogy.
Okay?
Imagine stretching a rubber band.
Okay.
When you let go,
Yeah.
it snaps back to its original shape.
Uh-huh.
That's elasticity in action. Okay, I can picture that. But how does elasticity apply to tissues in our bodies?
Many tissues, like tendons, ligaments, and even skin, have elastic properties.
Okay.
They can deform under stress,
Mhm.
but they return to their original shape
Mhm.
when the stress is removed.
So they're like biological rubber bands.
Yes.
Wow.
But different tissues must have different levels of elasticity, right?
Oh, right. That makes sense.
And that's where the concept of elastic modulus comes in.
Okay.
It's a measure of how stiff a material is
Yeah.
or how much it resists deformation.
So a tissue with a high elastic modulus would be very stiff,
Yes.
like bone,
Yeah.
while a tissue with a low elastic modulus would be more flexible, like cartilage.
You got it.
Okay.
And understanding the elastic properties of tissues is crucial. Everything from designing artificial implants to treating injuries.
Okay. I'm starting to see how this all connects.
Good.
But tissues don't always behave perfectly elastically, do they?
Sometimes they can be permanently deformed.
Think about what happens when you stretch a rubber band too far.
Yeah.
It might not snap back completely right.
Right.
It's entered the plastic region,
Okay.
where the deformation becomes permanent.
So tissues can experience both elastic and plastic deformation.
Exactly. And every material, including biological tissues, has a break-in point called ultimate tensile strength.
Okay.
It's the maximum stress it can withstand before it fails.
Wow. That's pretty intense.
It is.
I'm starting to think about all those sports injuries I've
Yeah.
seen where tendons and ligaments have been stretched too far.
That's a perfect example.
Yeah.
Understanding the mechanical limits of tissues is crucial for preventing and treating those types of injuries.
Okay, so we've got elasticity,
Yeah.
stress, strain,
Uh-huh.
and even breaking points.
Right.
But what about those weird materials that are both solid and liquid-like?
Yeah.
I remember learning about something called viscoelasticity
Yes.
in college,
Yeah.
but I'm fuzzy on the details.
Viscoelastic materials are fascinating.
They are.
They exhibit both viscous, fluid-like, and elastic, solid-like properties.
Okay.
It all has to do with how they respond to stress over time.
Okay, give me an example.
Think about honey.
Okay.
It flows slowly, like a liquid, but it also has some springiness to it.
Right.
That's because it's viscoelastic.
So it's like a combination of a solid and a liquid.
It is.
But how does this apply to biological tissues?
Many tissues in our bodies are viscoelastic.
Okay.
Cartilage, tendons,
Okay.
even blood vessels,
Right.
they all exhibit this time-dependent behavior.
Okay, my brain is starting to hurt a little.
I-
Can you break down some of these
Right.
time-dependent properties? What does it mean for a tissue to be viscoelastic?
Well, for example,
Okay.
viscoelastic materials can creep. That means they slowly deform over time under constant stress.
So if you apply a constant force to a viscoelastic material,
Yes.
it'll continue to deform,
Yeah.
even if that force is relatively small.
Precisely.
Wow.
And they also exhibit stress relaxation.
Okay.
That means if you stretch a viscoelastic material
Yeah.
and hold it at a constant length, the stress within the material will gradually decrease over time.
So it's like the material is relaxing
Exactly.
under that constant strain.
And another key property is hysteresis.
Okay.
That means the loading and unloading curves
Okay.
for a viscoelastic material
Uh-huh.
are different.
Hold on, I need to visualize this. Are we talking about like a
Sure.
graph of stress versus strain?
Yes.
Okay.
Imagine plotting stress on the y-axis
Yeah.
and strain on the x-axis.
Okay.
For a perfectly elastic material,
Yeah.
the loading and unloading curves would be the same.
Right.
But for a viscoelastic material,
Yeah.
the unloading curve will be below the loading curve, forming a loop.
Got it.
That loop represents energy lost
Okay.
due to the viscous behavior.
So it's like the material is remembering its deformation history.
Exactly.
That's pretty wild.
It is.
Yeah.
And understanding viscoelasticity
Yeah.
is crucial for designing biomaterials,
Okay.
understanding how tissues respond to injury,
Okay.
and even developing new treatments.
Okay. I'm really starting to appreciate
Yeah.
how much physics
He's a mix.
is involved in understanding how our bodies work.
It's all interconnected.
From the molecular mechanisms of muscle contraction
Yes.
to the viscoelastic properties of tissues.
Exactly.
It's all interconnected.
And we've only just begun to explore the fascinating world of biophysics
Right.
and its applications in medicine.
Well, I'm ready for the next chapter of our deep dive, but I think I need a quick break to let all this information sink in.
Take your time. We'll pick up with our final exploration when you're back.
Welcome back to the MedPod.
I feel like we've journeyed through, the microscopic world of our bodies.
We have.
From separating molecules in a lab
Yeah.
to understanding how muscles contract.
Right.
And tissues stretch and bend.
It's been quite a deep dive into the biophysics that underpin so much of what our bodies do.
Absolutely. But there's still one area I'm really curious about.
Okay.
Imaging. We've talked about how biophysics helps us understand things at a molecular level.
Right.
But what about seeing inside the body? I know there are techniques like X-rays and MRI.
Right.
But I'm particularly fascinated by ultrasound.
Oh, yeah.
It's amazing how sound waves can create images of our internal organs.
Absolutely.
And it seems much less intimidating than, say, being slid into a giant magnet.
It is. Ultrasound is a remarkable application of physics in medicine.
Yeah.
It's non-invasive, safe, and incredibly versatile. And as you mentioned, it relies on sound waves, but not the kind we hear. So we're talking about sound waves beyond the range of human hearing. How does that even work?
It all starts with a device called a transducer.
Okay.
This little gadget contains a special material called a piezoelectric crystal.
Piezoelectric. That sounds complicated.
It's actually a pretty simple concept. Piezoelectric materials have this unique ability to convert electrical energy into mechanical energy and vice versa.
Okay, so when you apply electricity to the crystal,
Yes.
it vibrates.
Exactly. And those vibrations generate the ultrasound waves.
Oh, wow.
Which are high-frequency sound waves beyond the range of human hearing.
Okay.
The transducer then sends these waves into your body.
So it's like a tiny speaker sending out sound waves that we can't hear. But how do those waves create an image? It's not like they have tiny cameras attached, right?
No tiny cameras. But the principle is similar to how bats use echolocation.
Oh.
The ultrasound waves travel through your body and encounter different tissues, like organs, bones, and fluids.
Yeah.
Each tissue has a different density, an acoustic impedance, which is basically a measure of how much resistance the sound wave encounters.
So when the sound wave hits a boundary between two different tissues, what happens?
Some of the wave is reflected back to the transducer, kind of like an echo.
Okay.
And since the transducer can also act as a receiver, it detects these echoes and converts them back into electrical signals.
So the transducer is like a speaker and a microphone in one.
It is.
But how do we get a picture from echoes? It seems like we just have a jumble of sound data.
That's where some clever physics and computing come in.
Okay.
The ultrasound machine measures the time it takes for each echo to return to the transducer. Knowing the speed of sound in tissue, it can calculate the distance to each reflecting boundary.
So it's like a super fast, highly precise sonar system mapping out the internal structures based on the echoes.
And by processing these echoes, the machine creates those black and white images you see on the screen.
Oh, okay.
The brighter areas represent tissues that reflect more sound, and the darker areas represent tissues that reflect less.
That's pretty cool. It's amazing how we can visualize the inside of the body without even making an incision. But I've also heard about Doppler ultrasound, which is used to measure blood flow. Is that the same type of ultrasound?
It uses the same basic principles, but Doppler ultrasound takes advantage of a phenomenon called the Doppler effect.
Okay.
You've probably experienced this with sound you can hear.
Like when an ambulance siren changes pitch as it passes by.
Exactly. That's the Doppler effect. The same principle applies to ultrasound waves.
Okay.
When the sound wave hits moving blood cells, the frequency of the reflected wave is shifted, either higher or lower, depending on whether the blood is flowing towards or away from the transducer.
So by analyzing this frequency shift, the machine can tell us the speed and direction of blood flow in different vessels.
Yes.
That's incredible. But I thought ultrasound was just for imaging. Aren't there also therapeutic applications?
You're right. Ultrasound isn't just for seeing, it can also be used to heal.
Okay, how does that work?
Therapeutic ultrasound uses higher intensity sound waves. And these waves can generate heat deep within tissues. It's like a very targeted deep tissue massage.
So the sound waves are like tiny little masseurs working on your muscles and tendons.
That's a good way to think about it. This deep heating can be used to relax muscles, increase blood flow, and promote healing. Physical therapists often use therapeutic ultrasound to treat conditions like tendinitis and muscle strains.
Wow. So ultrasound can see inside our bodies and even help heal them. It's truly a remarkable tool, and it's all thanks to a deep understanding of physics and how sound waves interact with biological tissues.
It's a perfect example of how biophysics is not just an academic pursuit, but a field with real-world applications that are improving healthcare and enhancing our lives.
Well, this has been an incredible journey through the world of biophysics.
We have.
We've covered so much ground. And I feel like I've gained a whole new appreciation for the intricate workings of my own body.
It's been a pleasure diving into these fascinating topics with you, and who knows what future breakthroughs await us as we continue to explore the interface of physics and biology.
I can't wait to find out. But for now, a huge thank you to all our MedPod listeners.
Thank you
for joining us on this deep dive into the incredible world of biophysics. We'll catch you next time for another fascinating exploration, the science behind health and medicine.